This video is part of a series that reviews the growth and development of the skeleton. Your objective in reviewing this video is to understand the origin, morphology, and function of the osteoclast. The core elements of this video are photomicrographs, each stained with hematoxylin and eosin, prepared at the Armed Forces Institute of Pathology in Washington, D.C. Despite the passage of time, the underlying original concepts, published and promulgated by Drs. Lent Johnson and Don Sweet from that institution, remain valid. Both are now past, and the video series adapted from their work honors them. Meeting these objectives should allow you to retain the content for long-term memory. 56 years ago, Lent Johnson stated, "Bone is not a tissue or a material, but a complex organ." A pathologist and biochemist, he painstakingly reviewed the osteoclast, the multinucleated giant cell pictured above, sitting in a sharp-edged cavity, a Howship's lacuna, the site of bone deletion. Bone consists of two phases, living cells and inert inorganic tissue called osteoid. The image is an H&E slide of cortical bone, stained pink by the eosin. 98% of the specimen is mineralized bone, and 2% is small, clear spaces called lacunae. The cells are osteocytes living in the lacunae. Osteoid is mostly collagen 1 that remains unmineralized for 3 weeks, undergoing a complex chemical preparation. Then, in a kerchunk moment, mineralization by crystals of calcium hydroxyapatite abruptly appear, filling 70% of available sites. As a living organ, bone is the primary structural element of the body, giving each person form. Its roles are locomotion, protection, a storehouse for hematopoiesis, and mineral homeostasis. The osteoclast is a motile, energy-packed, multinucleated giant cell, shining brightly for a day and then gone. The Howship's lacuna a sign of its presence. Note multiple, similar nuclei with adjacent clear spaces, digestin vacuoles containing material for deposition to the extracellular space. Pathologists say that if you see one osteoclast on an H&E slide, then significant bone deletion is occurring. The osteoclast arises from the marrow pool. The common myeloid progenitors, CMPs, of the monocyte macrophage lineage. These are basically uncommitted monocytes. Adjacent cells of osteoblastic lineage produce two markers: macrophage colony stimulating factor, M-CSF, supporting proliferation of CMPs, and RANKL, the master regulator required for osteoclast differentiation, survival, and activity. Unresolved research continues because many cells also express RANKL in inflammatory, neoplastic, and traumatic situations, leading to regional osteopenia. The short lifespan of the osteoclast and its efficiency, deleting 3 times its cell volume in one day, is offset by the requirement that it takes 3 months and 100 to 150 osteoblasts to refill that space. This disconnect between deletion in days followed by a fill-in over months leads to clinical situations wherein regional bone loss is so driven that there is risk of fracture. Clinical examples include acute overuse, acute infection, and rapidly growing tumors. These are the key references. Teitelbaum's paper will be discussed at length subsequently. The last reference, a complete overview of the advances in osteoclast biology, prompted this update to our original video on the osteoclast. As you read this last paper, Bear in mind Dr. Johnson's comments in 1964. He cautioned us on the interpretation of small animal research. In his words, the cortex of guinea pigs, rats, and rabbits is less substantial than many cancellous bone trabeculae of man, and may not give reliable data on the behavior of the human cortex. Most of the research work on the osteoclast has been performed on mice. For most of the videos, the emphasis is on what is occurring on the H&E slide. This video is more about the cell biology of the osteoclast. This diagram, adapted from Teitelbaum's work, summarizes the take-home message for this video. The cells involved on the left in osteoclastogenesis are stromal cells of osteoblast lineage resident in bone, osteoclast precursors in the monocyte macrophage family, and the osteoclast. The steps involved are the stromal cells produce cytokines RANKL and M-CSF, which recognize their receptors RANK and CFMS respectively on the macrophages, which are usually of marrow origin. The macrophages are thus stimulated to aggregate and fuse, and fully 10 to 20 cells become the mature osteoclast. The steps are blocked by osteoprotogerin, also produced by the stromal cells. The mature osteoclast is activated by RANKL to become active and delete bone. It changes to do this by polarizing on its contact with bone and by altering its cytoskeleton in response to matrix-derived signals which are linked to the integrin alpha V beta 3, leading to its ability to create a highly acidic microenvironment in the gap between it and the bone surface, the so-called resorption vacuole, where the dissolution of mineral precedes the lysing of the organic components of bone, principally by cathepsin K. The acidic environment in the resorption vacuole is created by the following: for the hydrogen ions, production inside the cell by carbonic anhydrase 2, and then transmembrane delivery via an H+ ATPase pump. For the chloride, delivery into the cell in exchange for bicarbonate via an electroneutral chloride bicarbonate exchanger, and then transmembrane delivery by an ion channel charged coupled to the H+ ATPase. The essential pathway for osteoclastogenesis converts the RANKL signal to changes in gene expression as follows. Osteoblastic lineage cells produce RANKL. On the monocyte, RANKL binds to RANK, a surface receptor. Within the monocyte, RANK recruits TRAP6, tumor necrosis factor, receptor-associated factor 6, leading to nuclear factor kappa beta activation and translocation to the nucleus. There, NF-kappa beta increases CFOS expression. CFOS interacts with nuclear factor of activated T cells C1, leading to transcription of osteoclastogenic genes. And with this, the stellanova is born. Osteoprotogerin protects the skeleton from excessive bone resorption by blocking RANKL. The balance between RANK and OPG is an important determinant of bone mass and integrity. What is the clinical relevance? The idea is to stick to principles and simplify. Generalized osteoporosis is common. The differential diagnosis is overwhelmingly increased clast activity, and the causes are multifactorial. The only condition wherein osteoblasts are defective is congenital and rare, osteogenesis imperfecta. Regional osteopenia is associated with cancer, inflammation, infection, and trauma. This is a result of enhanced expression of RANKL and M-CSF induced by local cytokines elaborated by tumor cells, local lymphocytes, and other sources, an area of active research. Disorders with increased bone formation are rare. They represent decreased clast activity. Osteopetrosis, or marble bone disease, presents as a result of various defects in the RANKL-OPG signaling axis, fully documented in Ono's recent reference. Adult Paget's disease is a condition of disordered remodeling that may present with increased bone formation. The juvenile form of Paget's presents with OPG deficiency. Both are anomalies with a spectrum of genetic defects. Bone removal can also be chemical, related to acid-base metabolism, or to oncosis. Oncosis is a chemical dissolution of bone around a dead or dying osteocyte. The result is slight enlargement of the lacuna, the space surrounding the living osteocyte shown in the scanning electron micrograph on the left. Bone acts as a mineral reservoir, supporting acid-base metabolism in an ongoing chemical-mineral exchange from its free surfaces. From the single lacuna, as shown on the SEM, several canaliculi emanate. The complexity of the combined lacuno-canalicular array can be imagined from the electron micrograph on the right. In this, the bone surrounding the osteocytes and dendrites has been dissolved. The remaining surfaces are around each vessel canal, the exposed surfaces of all trabeculae, and the endosteal layer of the cortex, a huge combined surface area of about 1,200 square meters. These surfaces provide a pool of ions, a ready resource for acid-base metabolism, about 72 hours worth. There is no deletion of bone involved whatsoever, but rather a give and take of ions of calcium, phosphate, and hydrogen from a thin, unmineralized layer. In the following H&E slides, I will outline in more detail the actual images of what I described in the opening diagram. Activated osteoclasts are found throughout the skeleton, often at the junctions of Haversian and Volkmann's canals. When stimulated by RANKL, they initiate bone deletion. Joined by other activated cells, they fuse and become multinucleated and fully functional. The cell polarizes and undergoes internal change, altering its cytoskeleton, forming a tight junction at the raw bone surface. 3 zones emerge: a sealing zone with complex biochemistry involving integrins, especially alpha V beta 3. Imagine the structural details that have to occur for this cell to move as it does along the bone surface as it fulfills its task of bone removal. A ruffled border, increasing the surface area for absorption, develops on the side opposite the raw bone surface, and a basal membrane on the outside isolates the clast from its surroundings as it begins its work. This creates at the bone surface a resorption vacuole. Once the clast starts resorbing bone, a 4th zone, a functional secretory domain, forms in the basal membrane, associated with the transport of material to the extracellular space. Mineral dissolution of bone occurs first under the ruffled border through the action of hydrochloric acid at a pH that is reported somewhere between 2 to 4.5. Cytoplasmic or carbonic anhydrase 2 drives hydrogen ion formation, which is then driven across the ruffled border by the H+ ATPase pump. The chloride ion has entered the cell in exchange for bicarbonate and moves to the ruffled border and into the resorption vacuole via a channel, charge coupled to the H+ ATPase, thus generating the hydrochloric acid which produces the required ambient pH. The acidity mobilizes the mineral phase, exposing the organic matrix of bone. The cell is filled with multitudes of mitochondria which provide the ATP for the significant energy required for bone dissolution. The proton pump is an absolute requirement for bone resorption to occur normally. Dissolution of the mineral leaves exposed the organic components, primarily collagen 1. Proteolytic enzymes, primarily lysosomal protease, cathepsin K, undertake the removal of these components. Inactivating mutations of the chloride channel are among the many defects in osteoclast function that cause human osteopetrosis, whereas the sclerosing bone disease pycnodysostoses reflects failure to produce functional cathepsin K. Tartrate-resistant acid phosphatase is also released into the resorption vacuole. The precise action of this enzyme is unknown. It does, however, leak out into the serum, leading to its use as an indicator of excessive bone resorption, a marker in metabolic bone disorders. The released collagen and other fragments, as well as solubilized calcium and phosphate, are taken in by endocytosis from the resorption lacuna into the cell. They're transported to membrane-bound vesicles within the cytoplasm, and you can see them here within the slide. There are lots of them. The degradation products are processed within the osteoclast, ending up in the specialized region of the basal membrane called the FSD. Ultimately, they are released into the extracellular space. The fate of the osteoclast is apoptosis. Little is known about how exactly this happens. This summary completes the video on the osteoclast. Use it as a touchstone to verify your recall of the concepts that you have just reviewed. Review the references, which provide much more detail on control mechanisms and the use of manipulating the RANKL OPG signaling axis to achieve improved treatment of metabolic and inherited bone disorders. We end with final acknowledgement of Dr. Sweet and Dr. Johnson. The video on the ultimate multitasker, the osteocyte, is next.